So our next series of speakers are going to be focusing on neurodegeneration. And the first speaker in that cohort is going to be Bill Rooney, who's a team with Jeff Ill from the Oregon Health and Science University. Bill?
Well, thank you. This is it's wonderful to be here. This is one of my favorite meetings of the year, and it's great to catch up on all the exciting work that's going on. I too am very saddened by the loss of Paul Allen. You know, at this particular time in history, having somebody that is generous, curious, and an informed critical thinker was more important than ever, and it's a real loss, and I'm very sorry for it. But his legacy and what he's set up, I think, is going to be exceptionally productive for many years to come. What I'm going to describe is some of our work on measuring glymphatic function, or following up on this physiology in human brain. And this is work done with Jeff Iliff and a team at Oregon Health and Science University just down in Portland, so just down I-5 a little bit. So just to remember, what the glymphatics is really a system of brain fluid dynamics. The brain is, of course, energetically very expensive to run. It's doing a lot of work. It's extremely tightly packed and organized as many tissue. And being so energetic, it's producing a lot of waste proteins, things that do have to be cleared. The lymphatic system within the parenchyma is not really well defined. So the idea of the glymphatic is that this highly organized tissue can accommodate brain water dynamics with organized flow. And it's really a mechanism to exchange the cerebrospinal fluid and the interstitial fluid. It's facilitated by perivascular flow and CSF conduction. One of the really interesting things is that sleep is thought to be involved in this, and that's one of the things we're going to explore. And during certain phases of sleep, there's a reduced parenchymal impedance, and that's due to a increase in the extracellular volume fraction and due to a volume shrinkage of certain cells. So something that's very exciting, but looking at how this physiology works in humans and extending some of the work that Jeff has done in small animals is important. But the goal of this work is to develop an approach to investigate this physiology in humans. So this is a slide from Jeff's early work using optical microscopy. It's beautiful. You can see how this tracer is distributing through brain tissue. Of course, it's an optical technique. It's a bit invasive and probably a little bit too invasive for what we want to be doing in humans. So great for animal studies, but not readily extendable to humans. So we've got to look for other approaches. And one approach that is a little bit less invasive is to not use a fluorescent dye, but use a paramagnetic dye and MRI to then follow how this paramagnetic compound moves through brain tissue. So this is a movie collected on our 12T instrument down at OHSU after an intrathecal injection of one of these paramagnetic dyes. And then you can characterize how things are moving through tissue at this kind of macro scale and get a lot of information about function of this physiology. But of course, these brains are pretty small. And really what we want to do is look at the human brain, which is much more massive than the mouse brain, the rat brain. And just making this transition between these, spatial scale is very important. Organization, the amount of white matter, the amount of cortical is very important. White matter distribution in the rat and the mouse, very different than in primate brain. So we're very fortunate at OHSU to have a primate, one of the natural primate centers associated with our facility. There are over 5,000 macaques at that facility. There's an aging colony. We're interested in characterizing how this physiology changes across the lifespan. So an important bridge that we've made use of is looking at the non-human primate and how some of these more invasive measures can characterize this physiology. And that gives us a link to validate some of the non-invasive measures and to better appreciate what's going on in a brain that's more relevant to the human brain. So this is one example. This is an overlay between a intrathecal injection of one of these low molecular weight, about 600 Dalton paramagnetic contrast agent in the non-human primate brain, combined with the magnetic resonance angiography. So you can see major arteries, major veins, and you can see that these tracers are really going in along these perivascular routes, and that's what was observed in the rodent brain, and we're able to do that here. This is characterizing the influx, the kinetics of influx of this small molecule into the brain. But it's important to be able to visualize because it's thought that pulsation and vessel pulsation is an important physical mechanism that's moving the tracer into the brain and through the brain. So being able to visualize that is really important. We can characterize, of course, these are all three-dimensional images. We've got some very nice imaging hardware out at the Primate Center. We can utilize that to characterize. The kinetics of this process actually are pretty prolonged, and that's a challenge, but we've got some really innovative ways to get around that. But this is just showing the distribution of this contrast agent in a relaxographic imaging space. Okay, and you can characterize that. As I said, they have an aged colony that Steve Kohama, who's a colleague, runs. We got some follow-up funding to look at this in non-human primates. And this is just now comparing how these tracers wash in, this is whole brain, in the young animal compared to the old animal. So monkeys in the wild live to about 22. At the Primate Center, they go into their 30s, a bit long. But that's a 27-year-old monkey is an old monkey. It's about a 3 to 4 year age scaling factor between humans and the macaques. But you can see gray matter and white matter, which has not quite taken up all of the agent at this time point, is characterized. But we're seeing increased uptake in the young animals, and that's really something that we're very interested in following up on. But now in moving to humans, we've talked about doing some of these intrathecal, and some groups in the world have done it. Usually it's in the context of a therapy or a diagnostic. We're very interested in what's going on in human subjects. Some of our teams, some of our neuroradiologists are all on board in doing this. I'm not so, I think staying less invasive is a better approach. So that's the tack that we followed. And one of the promising ways is to look at brain lactate as a surrogate. Lactate is, of course, a small molecule important in cellular metabolism. It turns out that lactate has a pretty high concentration in the extracellular space. And that's the space that we're really interested in characterizing. Of course, the Magistretti hypothesis, astron neuron lactate shuttle, there have been a number of rodent studies going back 50 and more years that show that brain lactate is decreased during sleep, though the mechanisms and the physiology for that is not really well described. And one of the things that we liked is that lactate can be measured non-invasively using a magnetic resonance technique, and that's one of our techniques of choice. This is just showing, this is a 3T instrument with one of our technologists who's been working closely with us on this project. So magnetic resonance spectroscopy. I show a spectrum here. This is frequency, amplitude here. You can see a couple of peaks. We can collect this from a relatively large area in the large volume in the parietal area. And you can see multiple peaks here in this spectrum. So we can collect a spectrum like this about just every 2 seconds or so. And temporal resolution is important, right? Because we want to characterize how things are changing in a time series. So in vivo NMR is good for characterizing brain chemistry. This is an example of a spectrum here. It runs on mostly standard MRI hardware. This is a pretty advanced instrument, but it's pretty easy to roll out to other sites. We've got really good time resolution, good signal to noise. One of the other things, MRI instruments, as I've mentioned before, they're not always the quietest places. So if you wanted to sleep, it might not be the most conducive environment for that. And these MRS techniques have low RF specific absorption. Safety is very important to us. We're putting a lot of metallic elements in that are very close to the subject. We want to make sure that everything is done safe. And these techniques are modular, adaptive, and we can really push them. They've been around for a while, but we can do different things like diffusion weighting on them. increased lipid suppression. We're interested in lactate, which is this small signal out here. And we can also push them to different nuclei to answer different types of questions. There's just a metabolic pathway. Of course, lactate is here. It converts to pyruvate through lactose dehydrogenase. There are monocarboxylic transporters that move lactate in and out of the cell. And of course, very important in overall cell metabolism. So this is work going back 1948, where these investigators were interested in seeing what the lactate levels were in the awake versus sleep. So I've replotted it here. And you can see a really marked change in lactate over these conditions. And this work has been replicated over the years. And most recently, by a couple of groups, this is showing how the extracellular lactate in mouse brain across sleep. This is awake state, and it goes into a sleep state. You can see that, so the kinetics of lactate, it's a time constant of 10 or 15 minutes to wash out of that extracellular space. And you can see as the animal's cycling, going through sleep and wake and then sleep states, you can see this signal cycling. So nice work that was done relatively recently. Last year, Megan Nedergaard's group published this work showing brain lactate as a biomarker, and this is in the mouse. And here you can see brain lactate in the dark phase when the animal is awake, and then in the light phase when the animal's sleeping, there's this substantial decrease. The group did manipulations to modify the CSF dynamics, and you can see in most of these perturbations you have a much muted change in lactate compared to control state. And one of the other interesting aspects is that they measured how lactate was moving out of the brain through the lymph nodes. And you can see that there's a lot of lactate moving out in the normal animals. When they tie off those, there's much less. So again, this is really very supportive of the idea that lactate is a possible surrogate for this physiology that we're interested in. So part of this is, as I mentioned, we do have to characterize sleep in the MRI instrument. We do that by, here's Jeff, he's looking pretty happy with this network of electrodes around his head. It takes a bit of time, about an hour to prep this and get all of the electrodes. It's a low density electrode. We don't need a lot to characterize sleep. In my view, more is better here. So we modified our MR instrument and our pulse sequences to work easily and reliably with this hardware. And of course, we spent a lot of time assessing safety because we're putting, MRI is really a benign imaging technique. It doesn't use ionizing radiation, but we're putting a lot of power into that instrument. And that power can have really, really bad consequences if you're not careful. So we do pay a lot of attention to safety and characterize it. This is just showing some of the EEG that we pulled out while the subject was in the scanner. If you were to look at it in an unfiltered approach, I mean, the coupling between the MR scanner and the EEG is tremendous. So these are really pretty nice EEG. And our team can read them and characterize them. And they do that across 30-second epochs. Here again is a spectroscopy example. So we collect something like this every 2 seconds. We can then average across different states. And we do that by, again, having the subject, our champion subject was in just over 3 hours, which is pretty amazing. We weren't even sure we'd get people to sleep in the scanner, but we do a brief overnight sleep deprivation in our young healthy, and then they actually fall asleep pretty quickly, and they follow this kind of a, what's called a hypnogram, just characterizing when they're awake in different stages of sleep. And then we look at that during this type of time period, so they'll cycle through a few different phases. We then can, since we have very high temporal resolution with our MRS measure, we can rebin according to which sleep state they're in, and then do all of our analyses. And this is an example of brain lactate levels for a 27-year-old healthy young female subject. And here's a hypnogram. So you can see this is like textbook. We'd really like to see this. A long period when the subject was awake, and then they begin cycling into sleep. And they're spending a fair amount of time here in a deep sleep, in an N3, a non-REM stage 3 sleep. And then toward the end, they come back out. And you can see the lactate concentration, how that's changing in time. And we're seeing in this subject a pretty meaningful decrease in lactate concentration in the deep sleep state. And this is one individual, right? So that's great. If you can do this in an individual, that's a pretty powerful approach. we've done that now in a group of young healthies, and this is the group's aggregate summary. And again, we're seeing this change in lactate concentration. We pull out about just over 600 micromolars of lactate in the brain, and it's going down by about, on average, about 25% at the N3 sleep state. And these are young healthies. We've started doing this in a group of older individuals, and surprisingly, we can get them to sleep in the scanner as well. The EEGs are pretty good. They're not quite as good. The impedance is a little bit different. But what the early data show are that we're seeing a reduced, kind of a muted response in the change in lactate, which is really interesting, and we're following up on that now. In fact, there's an older subject in the magnet right now as I'm talking. One of the other things that we were able to do is to put some modules on the spectroscopy sequence, put diffusion modules. And this just shows apparent diffusion coefficient for different metabolites that I indicated in that magnetic resonance spectroscopy plot. And this is N-acetyl aspartate, choline, total creatine. So these are thought to be primarily intracellular metabolites, NAA localized really to within neurons within the brain. And here's lactate. So what we're seeing with lactate is a very high apparent diffusion in the awake and N1 state. And here we've grouped the N2 and N3 in time state 2. So we see a big change in lactate diffusion properties going between these states, and not much going on with the other metabolites. So lactate is kind of behaving in this sleep state more like these intracellular metabolites. So two things happen. We lose concentration, and the apparent diffusion changes markedly. And it's not conclusive that but it's really supportive of this concept of glymphatics, and that is that lactate in the extracellular space is being washed out, and it's a selective population that's being washed out. So then, to summarize, so proton MRS detects roughly a 25 to 30% decrease in brain lactate in sleep for young healthy individuals. We see these apparent diffusion properties of lactate signal change markedly in sleep. And these findings support that there's a selective pool of lactate, or a pool that's selectively reduced. And we think that's the extracellular lactate pool. We've got some other experiments that we're going to be doing to validate these. But the preliminary data also suggests a smaller change in the lactate during sleep in older healthy individuals, and we're following up on that now with an older cohort. One thing I didn't talk about, what's interesting, we didn't see any significant change in water diffusion properties in this, which are kind of interesting. Other work, we've also looked at sodium-23. With the idea that if there is an expansion of the extracellular space, we might see an increase in that sodium signal. We did not see that, which is kind of interesting, but it could simply reflect an ion redistribution within cells, intracellularly, extracellularly, that's kind of explaining. So we will follow up on that as well. So these approaches are really safe and well tolerated in healthy individuals from 21 to 81. The non-human primate studies are really important. They do, since we have that age cohort, suggest reduced tracer uptake with age. Again, this is following this whole concept that age is a major risk factor for neurodegeneration, and the glymphatic physiology may be a very early marker for who's at risk for neurodegeneration. And access to the non-human primate is, of course, a very valuable tool to validate. So I'll end here and thank all of the people that made this happen. Selda Yildiv, who's a postdoc that did a lot of the work, Manoj Sami, Catherine Powers, Miranda Lim, characterizing the EEG, and then a whole host of other individuals that contributed in important ways. And of course, the funding came from the Paul G. Allen Frontiers group, and I'd like to thank them for supporting us. And there's actually been a lot of follow-up work that's come out of this now that I think is really, it's kind of launched 2 postdoc careers, which is great. There's been a lot of follow-up funding that's been applied for bringing other groups together, and it's all very, very powerful and impactful. And thank you for your attention.
We have time for a question or two. The microphone's coming to you. Or you could try shouting.
Yeah. I can get close.
There you go.
Hi, it was nice. Your lactate, the decrease with sleep, you showed it for the young population, and you say you're doing it for the old. Do you have any inclinations to whether the lactate decrease is less in the, or disturbed in the older population? So you see less
so two
change?
things we see from the older population. The resting level of lactate is a little bit lower. And the decrease during sleep is also lower. So if this idea of astrocyte neuronal coupling and just that metabolic couple, it could be that loss of this physiology is a astrocyte dysfunction. And it could be that some of these markers of low lactate, even in the resting state, could be a handle on understanding that. But it's really interesting. We're kind of building, we've only done 3 of the older group so far, and we're building up those numbers now.
Dave?
So I'm curious about your reluctance to use this in a theranostic strategy and why that is, because it seems like there's a credible opportunity, especially in the setting of intrathecal chemotherapy.
I'm sorry, for the intrathecal administration?
Yeah.
So there are a lot of camps on this, right? I happen to believe that if you select the right MRI contrast agent, it's probably fine. I've had many administrations of these agents for other types of, so there are some bad players out there. It's gotten a lot of bad press recently, and for good reason. I think there are real concerns. It's probably been overutilized, and there are just some bad actors, things that, compounds that are a bit more labile, that probably don't add a lot of value to the field. So I think it's preferred not to, in some settings, in some medical settings, certainly, but as a screening tool, it's not something that many people would jump and say, yeah, I'll volunteer for that. But we do, having the non-human primate is really important, because that's, to me, a much more direct measure of this CSF-ISF interchange, which is kind of that backbone of the physiology. So being able to characterize that and then do the same measurements we're doing in humans in a well-characterized state, just to get that validation, is really important in a brain that has very similar kind of distributions of gray matter, white matter, a little bit smaller, but still very, very important. Vasculature that's very much the same.
So actually it's time.